Hello and welcome again. Today we want to have a look at the diversity of African archaeobotany. As the second largest continent, Africa offers an extremely broad environmental and cultural diversity, resulting in a broad variety of different research topics to archaeobotanists. Africa has 8 major physical regions: the Sahara, the Sahel, the Ethiopian highlands, the savanna, the Swahili coast, the rainforest, the African Great Lakes, and southern Africa. Some of these regions cover large bands of the continent, such as the Sahara and Sahel, while others are isolated areas such as the Ethiopian highlands and the Great Lakes. Each of these regions has unique animal and plant communities. The etymology of Africa is greatly disputed by scholars. The origin of name is often related to the Phoenician, Greek, and Roman culture. The Egyptian word Afroika means motherland. The Greek Afrike is translated as without cold, and the Latin word aprica means sunny, so all of them seem to have some justification. As the cradle of humankind, Africa has a unique place in human evolution and the history of humankind, with fossil evidence of Homo sapiens and their ancestors through each key stage of their evolution. These include the Australopithecines, Homo habilis, and Homo erectus. Human movement or migration not only plays a role in understanding early phases of population of Eurasia, but is also key to the cultural landscape of Africa. Geoscientists and archaeologists are especially interested in migration as it relates to all kinds of evolution and can explain how social and cultural practices are spread throughout the world. Two later migration patterns, the Bantu migration and the African slave trade, characterize the cultural geography of the continent. The Bantu migration was a massive migration of people across Africa about 2,000 years ago. It represents the most important human migration to have occurred since the first human ancestors left Africa more than 1 million years ago. Lasting for 1,500 years, the Bantu migration involved the movement of people whose language belonged to the Congo-Niger language group. The common Congo-Niger word for human being is Bantu. The Bantu migration was a southeastern movement. Historians do not agree on why Bantu-speaking people moved away from their homes in West Africa's Niger Delta basin. They first moved southeast through the rainforests of Central Africa. Eventually, they migrated to the savannas of the southeastern and southwestern parts of the continent, including what is today Angola and Zambia. The Bantu migration had an enormous impact on Africa's economic, cultural, and political practices. Bantu migrants introduced many new skills into the communities they interacted with, including sophisticated farming and industry. The skills included growing crops and forging metal tools and weapons. These skills allowed Africans to cultivate new areas of land that had a wide variety of physical and climatic features. Many hunter-gatherer communities were assimilated or adopted into the more technologically advanced Bantu culture. In turn, Bantu people adopted skills from the communities they encountered, including animal husbandry or raising animals for food. This exchange of skills and ideas greatly advanced Africa's cultural landscape, especially in the eastern, central, and southern regions of the continent. Today, most of the population living in these regions is descended from Bantu migrants or from mixed Bantu indigenous origins. The third massive human migration in Africa was the African slave trade between the 15th and 19th centuries, but we will not discuss it here as there is no archaeobotanical research associated to it. Let's have a look now at the ecological zones of Africa, starting with the Sahara. It is the world's largest hot desert, covering 8.5 million square kilometers, making up 25% of the continent. The Sahara has a number of distinct physical features, including ergs, regs, hamadas, and oases. Ergs, that is sandy desert, which cover 20% of the Sahara, are sand dunes that stretch of hundreds of kilometers at heights of more than 300 meters. Ergs cover most of Algeria and Libya and parts of Mali and Nigeria. Ergs can contain large quantities of salt, which is commercialized today. Regs are plains of sand and gravel that make up 70% of the Sahara. The gravel can be black, red, or white. Regs are the remains of prehistoric sea and riverbeds, but are now nearly waterless. Hamadas are elevated plateaus of rock and stone that reach heights of more than 3,000 meters. They include the Atlas Mountains, which stretch from southwestern Morocco to northeastern Tunisia, the Tibesti Mountains of southern Libya and northern Chad, and the Ahaggar Mountains in southern Algeria. An oasis is a hub of water in the desert, often in the form of springs, wells, or irrigation systems. About 75% of the Sahara's population lives in oases, which make up only slightly more than 2,000 square kilometers of the desert's vast area. The Sahara's animal and plant communities have adapted to the extremely dry conditions. Saharan plants survive thanks to root systems that plunge as far as 24 meters underground. In some parts of the Sahara, such as the southern Libyan desert, plants cannot take root at all. Only few archaeobotanical projects are situated in the Sahara. Some of them are conducted in the Libyan desert by an Italian research team from the University of Modena, led by Anna Mercury. The researchers addressed questions of pastoralism in the Tadrart-Akakus Massif in southwestern Libya in the central Sahara by studying dung remains from the Takarkori rock shelter dating to the Saharan late pastoral Neolithic at about 5,700 to 4,650 calibrated BP. Applying means of palynology and geoarchaeology within a spatial approach, they found that the pollen trapped in the coprolites is a seasonal marker and suggests the rock shelter was occupied by Neolithic pastoralists in the dry seasons between late autumn and early spring, with possible variations year by year depending on specific resource availability. Moreover, the data from the excavation and micromorphological thin sections suggests repeated use of the same areas for panning. Insights on transhumance and mobility patterns can be extrapolated and generate an understanding about herd composition and their links to social organization in the mid-Holocene Sahara. There is a comprehensive body of paleoclimate and archaeological research investigating the so-called time of the Green Sahara. For those of you who are interested in more video clips on this topic, just visit some of the links in this channel. The Sahel zone is a narrow band of semi-arid land that forms a transition zone between the Sahara to the north and the savannas to the south. It is made up of flat, barren plains that stretch roughly 5,400 kilometers across Africa from Senegal to Sudan. The Sahel contains the fertile delta of the Niger, one of Africa's longest rivers, but the Sahel's fertile land is rapidly becoming desert as a result of drought, deforestation, and intensive agriculture. This process is known as desertification. The Sahel's green vegetation only emerges during the rainy season, but is often quickly harvested by farmers or consumed by animals. Baobabs are drought and fire-resistant trees with huge trunks. Acacia, whose deep root systems are ideal for semi-arid climates, are among the most common trees found in the Sahel. Krum, a prickly grass, is today the primary fodder of Sahel's herds such as Cebu cattle. The Sahel zone, together with Savannah, are major research regions for a large number of archaeological projects and have been extensively studied by archaeobotanists for the origins of food production, and we will hear later more about some major outputs of this research. The Ethiopian highlands consist of 80% of Africa's tallest mountains. The highlands' craggy landscape is perfect for nimble animal species. Native species such as the Valia ibex, an endangered wild goat, and the Gelada baboon live in the ledges and rocky outposts of the Simien Mountains. The most emblematic highland species is probably the Ethiopian wolf, which is now on the brink of extinction. Important plant species native to the Ethiopian highlands include the Ethiopian rose, Africa's only native rose, and the ensete, a tall, thick, rubbery plant that is a close relative of the banana. Archaeobotanical research has started only recently in Ethiopia by a research team around Michael Harrower from Johns Hopkins University in Baltimore at the Aksumite site of Beta Samati. Savannas or grasslands cover almost half of Africa, more than 13 million square kilometers, and make up most of Central Africa, beginning south of the Sahara and the Sahel and ending north of the continent's southern tip. Among Africa's many savanna regions, the Serengeti or Serengeti Plains is the most well-known. The Serengeti is a vast undulating plain that stretches from Kenya's Masai Mara Game Reserve to Tanzania's Serengeti National Park. The Serengeti is home to one of the continent's highest concentrations of large mammal species, including lions, hyenas, zebras, giraffes, and elephants. Each year, more than 1 million wildebeest travel in a circular migration, following seasonal rains across the Serengeti Plains, which fosters regeneration of grassland. Extended parts of this landscape have also been subject to archaeological and archaeobotanical research and will be discussed further down this track. But see also the works by the team around Katharina Neumann at the University of Frankfurt. The Swahili coast stretches about 1,600 kilometers along the Indian Ocean, from Somalia to Mozambique. The nearby coral reefs and barrier island protect the coast from severe weather. More vegetated areas are located on a narrow strip inland from the coastal sands. Heavy cultivation has diminished the diversity of plant species in this interior area of the Swahili coast. Mangrove forests are the most common vegetation. Mangroves have exposed root systems, and this allows the trees to absorb oxygen directly from the air as well as from the nutrient-poor soil. There are quite some archaeobotanical studies from the Swahili coast, mostly presenting results of phytolith analysis and conducted by a team of the Spanish National Research Council in Barcelona, headed by Marco Madelia. The researchers studied phytoliths from Songomnara, a stone town in Tanzania of the 14th to 16th centuries, which was built as one of many trade towns on the Indian Ocean. The joint UK-American project seeks to explore the urban space of Songomnara as an example of the structuring of that Swahili stone town. Fieldwork encompasses a range of different techniques aimed at understanding activities across the site and includes, among others, phytolith and paleobotanical studies. While the macrobotanical data are not yet published, a combined application of soil micromorphology, soil chemistry, and phytolith analysis could pinpoint the presence of sectors of possible stables near the houses, interspersed with what might have been garden plots or orchards, which indicate an articulated use of the open space between the buildings. As with many other studies that combine geoarchaeological methods and plant microfossils, the spatial organization of the population, in this case land use patterns for cultivating vegetable or fruit trees, represent a major research topic. Most of Africa's native rainforest has been destroyed by development, agriculture, and forestry. Today, 80% of Africa's rainforest is concentrated in Central Africa along the Congo River basin. Africa's rainforests have a rich variety of animal life, and plant communities are even more diverse, with an estimated 8,000 plant species documented. More than 1,100 of these species are endemic, that is, they are found nowhere else on Earth, and only 10% of the plants in the African rainforest have been identified to the current day. Particularly the more cleared areas of the tropical rainforest have been archaeobotanically studied. There are a larger number of research articles from the archaeobotanical team at the University of Frankfurt that contributed to our understanding of early millet cultivation and that are subject to one of the forthcoming presentations. The Great Lakes are located in 9 countries that surround the Great Rift Valley. As the African continent separated from Saudi Arabia during the Oligocene, large deep cracks were created in the Earth's surface and later filled with water, resulting in some of the largest and deepest lakes in the world. There are 7 major African Great Lakes: Lake Albert, Lake Edward, Lake Kivu, Lake Malawi, Lake Tanganyika, Lake Turkana, and Lake Victoria. Lake Victoria is the largest lake in Africa and the southern source of the Nile River. The African Great Lakes region has also a diverse range of aquatic and terrestrial animal life, such as the Nile perch, that has been identified in a number of Bronze and Iron Age sites in the southern Levant and was at that time a desired food resource in the Mediterranean trade system. The Great Lakes abut everything from rainforest to savanna plant communities. However, invasive species like the water hyacinth and papyrus have begun to take over entire shorelines, endangering animals and plants. As the Great Lakes region is also homeland to the later Eastern Bantu culture, archaeological and archaeobotanical focus was frequently on the development of Iron Age pastoralism in this region. Also, this region will be partially covered by the forthcoming presentation. The region of southern Africa is dominated by the Cap Vaal Craton, a shelf of bedrock that is more than 2.6 billion years old. Rocky features of southern Africa include plateaus and mountains, such as the Drakensberg Range, and southern Africa is the epicenter of Africa's well-known reserves, such as the Kruger National Park, which protects animal species such as lions, elephants, baboons, white rhinos, and Burchell zebras. Other important animal species include the impala, a type of deer, and the springbok, a type of gazelle that can spring several feet into the air to avoid predators. Southern Africa's Cape Floristic region is one of the richest areas for plants in the world. While the Cape Floristic region covers less than 0.5% of Africa, it is home to nearly 20% of the continent's flora. The giant protea, South Africa's national flower, is home there. Archaeobotanical research has been infrequently applied since the 1980s, and only few works are addressing farming communities, whereas a couple of studies have been conducted on Pleistocene occupation sites, such as the Middle Stone Age Sibudu Cave in the KwaZulu-Natal province. We will hear about this in one of the upcoming presentations. Having seen the enormous diversity in landscapes and environmental conditions, it is easily comprehensible that research topics addressed by archaeobotanists working in Africa are so diverse as are the climatic regions on the continent. Most of the regions are located in tropical climates, of which the majority is dominated by arid and semi-arid conditions. As you can see in both of these maps, they are not strictly climatically based, but both integrate in different ways vegetation zones and thus are not identical in the regions they depict. The terms we are using here for describing the specific types of archaeobotany are equally characterized by different aspects. For example, tropical archaeobotany usually relates to locations in humid environments, that is, for Africa, mostly countries with a certain portion of rainforest, for example, Congo, Gabon, Cameroon, and others. As preservation of plant macro remains in humid zones is very limited compared to temperate or arid regions, the scientific approach might vary from each other. We will hear more about tropical archaeobotany in the particular context of the Americas in one of the next lectures. Here we focus on the Sahelian regions and the dry tropics of Africa that include the different types of savannas. Most of the archaeobotanical research has so far been conducted in these regions. We will not consider typical Mediterranean archaeobotany, which also comes with a multitude of diverse research foci, and we will not treat Egyptian archaeobotany, which is on the basis of its cultural association with the Levant, rather belonging to Near Eastern archaeobotany. How much this is true shows the map, which unfortunately comes in a very bad quality. It, however, brings these essential differences to the spot. When looking at the earliest recorded cereals around 3500 BC, they are represented by wheat and barley along the Nile River, with the oldest finds in the north and younger finds in the south. The cultural background relates them to the early Bronze Age Levant, indicating the economic ties to these two regions from this time onward. In contrast to this, the typical sub-Saharan species of sorghum, finger millet, and pearl millet appear much later and in different regions, representing culturally and economically distinct areas. The archaeology of Africa is extremely rich throughout all periods of humans' biological and cultural evolution, and we cannot even touch some of the major cultural units and archaeological discoveries within this seminar. The aim here is to trace cultural development from the perspective of human-plant interaction, that is, how early humans made use of plant resources. Archaeological research of the 1970s and 1980s started to focus on regions other than Egypt, mostly on sub-Saharan Africa. It has been assumed since then that the origin of food production in the Sahel went back to pastoral communities of the Sahara and probably occurred in 3 phases, as pictured in the figure on the left. The first of these would have comprised the 2nd millennium BC and saw the massive immigration of pastoral communities from the southern Sahara and the establishment and flowering of settled food-producing communities in some core regions. One of the biggest unanswered questions at that time was whether the cultivation of pearl millet, that is Penicetum glaucum, as the first and only crop was a Sahelian innovation, or whether it was imported along with domestic animals by incoming Saharan pastoralists. In a 2013 publication, Peter Bräunig points out that evidence for agriculture outside Egypt goes back little more than 4,000 years, whereas the first domestic animals go back almost twice as far. Most important in Africa were cattle, followed by sheep and goats in different proportions according to time and place. The last 2 certainly originated in the Near East, but opinions differ as to the time and place of the domestication of cattle and the beginning of pastoralism as a livelihood in which domestic animals were at the center of subsistence. It was clear, however, that a pastoral economy in which cattle were critical was widespread over the central and southern areas of the then still Sahel-like Sahara by at least 5000 BC. By 2000 BC at the latest, the inroads of late Holocene aridity brought about the migration of Saharan pastoralists into the savanna south of the Sahara and the beginning of food production in the Sahel. The southern Sahara and the western Sahel are the homeland of the wild forms of important African crops such as pearl millet, Penicetum glaucum, and African rice, Oryza glaberrima. Along with East Africa, these areas have been considered since the 1970s to be one of the hypothetical birthplaces of Africa's crops. Today, about 50 years after Harlan's suggested areas of domestication, researchers can confirm and even specify Harlan's ideas with the means of archaeobotany. One of the most burning research topics in archaeobotany have therefore been since long the beginnings of domestication and cultivation of plants in Africa. The figure shows some selected crops of African origin that have been central to archaeobotanical research over the last decades, including cereals such as sorghum bicolor, with its hulled race of bicolor and the free-threshing dura race, pearl millet, Penicetum glaucum, and finger millet, Eleusine coracana. The pulse pods and seeds shown in the picture include the cowpea, Vinia unguiculata, hyacinth bean, Lablab purpureus, and Bambara groundnut, Vinia subterranea, and insect, Insete ventricosum, a banana species. The following information has been mostly extracted from Dorian Fuller's works. We already know that Africa's sequence of economic change differs from that of most continents, and that herding tended to precede crop farming, and domestication of indigenous plants occurred fairly late during the last 5,000 years. Different innovations in subsistence strategies took place in different parts of Africa. Some regions saw animal domestication and the spread of herding, others the domestication of seed crops or the cultivation of perennial plants propagated vegetatively. These distinct trajectories of change often show different causes, consequences, and processes for different transitions to food production. Although still sparse, solid archaeological evidence for these processes is beginning to accumulate in different parts of Africa. This evidence allows us to trace how the domestication of various indigenous African crops led to the development of a wide array of agricultural economies within Africa and, eventually, how those economies gave new resources and tastes to other regions of the world. The shift from procuring wild resources to food production can be considered as the most fundamental economic change that occurred in prehistory. It involves intervention in the life cycles of plants and animals to enhance their productivity, palatability, distribution, predictability, or other traits relevant to human consumption. The pathway to food production may differ in diverse geographic and sociocultural regions and is important for understanding the co-evolution of plants and humans. Although we will consider the origins of agriculture in the Near East in detail again in one of the following lectures, I want to describe shortly some principles that apply globally and that go along with the different pathways to food production. They are required to be understood to evaluate this major economic change and have been described first by Harris in 2007 as outlined here. They comprise the characteristics of cultivation, domestication, and agriculture. Cultivation is a human action that enhances the survival, reproduction, or growth of certain plants. It may include preparing soil, plant seeds, seedlings, tubers, or other plant parts, weeding and tending plants as they mature and/or protecting them from predators. Domestication describes genetic and morphological changes on the part of a plant population in response to selective pressures imposed by cultivation. Whereas cultivation denotes a change in human activities, domestication is an ensuing change in plant genotype or morphology. Most seed crops share a clear grain domestication syndrome of morphological change, but viticultural domesticates have diverse biological responses to cultivation. Agriculture is the form of land use that results from cultivation and/or domestication. It is a change in the landscape as people regularly cultivate and focus more attention on domestic plants. Agriculture creates fields for larger-scale production of crops. How one distinguishes agriculture from small-scale cultivation varies according to the parameters of particular geographic and cultural contexts. The shift to dependence may or may not be rapid or permanent. Some societies have existed for millennia in a state of low-level food production without crossing such a threshold. In the case of seed crops, one expects cultivation first, followed by domestication and finally agriculture. Southwest Asia's archaeological record follows this sequence. However, this sequence need not apply in every instance. Vegetatively propagated plants may be cultivated on a large scale without exhibiting permanent morphological or genetic change, and decrew techniques, that is, broadcasting seeds into receding floodwaters, could create optimal environments for wild populations of cereals. These evolutionary schemes imply several distinct transitions that must be identified and explained. Initial cultivation, a key behavioral change in humans, is difficult to discern. During pre-domestication cultivation, morphological changes begin to occur, some of which may be evident in assemblages of ancient plants. The shift to agriculture may be detected quantitatively via documenting cultivated species in subsistence remains. The clearest archaeological indicators of cultivation are phenotypically distinct domestication traits, such as the tough rachis that hold cereal grain securely. These traits may gradually rise in frequency in cultivated populations. Their predominance marks the end of a process of biological evolution, that is, the domestication, that began with an earlier change in human practices. This is cultivation. Other traits, such as grain size, evolve gradually under cultivation. Diachronic change may be evaluated in large assemblages with an extended temporal range, but isolated specimens are ambiguous. Determining wild versus domestic status for vegetatively propagated plants can be even more challenging. Once plant remains are identified conclusively as domestic, archaeobotanists must assess their quantity, contextual integrity, and consistency with other aspects of the archaeological record. As we have heard in one of the previous lectures, investigation of early crops and agriculture is conducted by different complementary data sources: modern botanical and ethnobotanical studies, and ancient plant remains recovered from archaeological or paleoecological sites. Modern botanical and genetic studies of crops and their sister taxa can assess what differences have evolved between domesticated and wild plants and identify which processes of natural or human selection might have fostered these changes. Ecological and geographical research focusing on wild progenitors, which are populations of wild plants closely related to domestic crops, can provide information on the regions and kinds of habitats where cultivation began. Linguistic studies can shed light on the past use of wild and domestic plants by various cultural groups and the pathways by which specific crops probably spread. Ethnographic and ethnobotanical observations of traditional cultivation methods and the use of wild progenitors and complementary resources can clarify the economic and social circumstances of prehistoric plant domestication. And of course, as we all know, ancient plant remains and the contexts in which they appear form another data source. Key aspects of archaeobotanical studies pertaining to plant domestication include morphological assessment of a specimen's wild versus domestic status, direct dating of ancient domesticates, and understanding the social, economic, and environmental contexts of early crop use. Macro remains, so seeds, fruits, and other parts like chaff, can furnish concrete evidence of early crops in agriculture. Charred seeds of crops such as pearl millet, finger millet, and cowpea allow us to study changes in size and/or shape over time. Preserved chaff can reveal other domestication traits, such as whether seeds were dispersed naturally or were processed by threshing and dependent on human sowing. Studying plants that are not harvested for their seeds, such as tubers and some tree fruits like oil palm, requires examining other lines of archaeobotanical evidence. Phytoliths, microscopic silica bodies deposited in plant tissues, may be used to identify inseed and bananas, because these members of the Musaceae family have distinctive volcano-shaped phytoliths. Charred fragments of some roots and tubers may be identified on the basis of their parenchyma tissue. Recovery and recognition of starch grains found in residues on ancient tools and ceramics may allow identification of yams or other tubers and other plants processed by ancient people. Phytolith parenchyma and starch research is just beginning to contribute new perspectives to African archaeobotany. Archaeological and botanical studies have revealed evidence for a growing number of distinct regions and independent domestication around the world. Among the 24 possible centers of early cultivation now recognized, at least 5 are in Africa. Botanically, the closest wild relatives of many crops have been identified as native to Africa. Knowledge of their modern geographic distribution, refined by genetic, systematic, and paleo-environmental studies, allows us to outline 5 regions of initial cultivation. The West African Sahara and Sahel has wild pearl millet, and clear archaeobotanical evidence for its domestication at least 4,500 years ago. Watermelons probably also originate in this zone. To the south, the West African grassy woodlands are home to fonio cereals, cowpea, bambara groundnut, African rice, and the baobab tree. It is unclear whether these were domesticated before or after domestic pearl millet spread into the area. Further south, in forest margin areas, Western African yams, oil palm, and kola nut were first brought into cultivation. East of Lake Chad, East Sudanic grasslands were the likely home of early cultivation of sorghum and perhaps the hyacinth bean. The Ethiopian uplands have several domesticates, the tiny seeded cereal teff, the oilseed nook, a local pea, and enset, which provides large quantities of starch and construction and wrapping material. Early cultivation of Eastern African yams, coffee, and anchovy, which has edible fruits, tubers, and greens, probably took place at slightly lower elevations. Finger millet domestication probably occurred in upland Ethiopia or other highland parts of Eastern Africa. And to conclude this lecture on African archaeobotany, I would like to draw your attention on the continuously increasing knowledge on early crops just 6 years later from this. Africa's most important contribution to global agriculture is sorghum, sorghum bicolor, which, remember, has been attributed to domestication somewhere in the eastern Sahelian savanna transition zone by Harlan already in the 1970s, based on geobotanical studies. Current genetic data and recent archaeobotanical reports support this argument and point to a domestication process that commenced before 3000 BC and was completed probably after 2000 BC. Current evidence, although still limited, suggests sorghum domestication did not have taken place as a consequence of pastoralism, because wild forms are associated with agricultural sites in the north, whereas domesticated forms occur in hunter-gatherer sites in the south. The domestication process of sorghum itself is still only known from limited data, but these do provide a basis for estimating the rate of cultivation and the evolution of domestication traits. Fuller calculates from the proportion of non-shattering versus wild-type spikelet bases that domestication rates must have been similar to those of barley, einkorn, and emmer wheat in Southwest Asia. This also contradicts older notions that sorghum domestication was slower than other cereals due to cross-pollination. Katharina Neumann was the first to criticize the hypothesis that African cereals were harder and slower to domesticate due to their tendency of cross-pollination. Sorghum spread rapidly as a crop, both within Sudan and beyond, potentially while even still undergoing domestication. As long recognized, sorghum has dispersed via maritime routes from eastern Sudan, Eritrea, to western India by the period of 2000 to 1700 BC. The current evidence implies that dispersal to India occurred before non-shattering was fully fixed in cultivated sorghum populations. Subsequent to this, sorghum must have spread through the savannas of Africa, although empirical data remains few. Pearl millet, Penicetum glaucum, was the earliest domesticate of Western Sahelian Africa and provides both parallels and contrasts to processes in sorghum. Also here we should note that the region of original domestication was already delineated through geobotanical works. As with sorghum, domestication can be inferred to have taken place in the later mid-Holocene, as the Sahara expanded and as pastoralism spread. Data from Mali and Mauritania provide evidence marking the end of the domestication episode in this species around 1300 BC at latest. Presumably, cultivation and the evolution of non-shattering had begun some 1,000 to 2,000 years earlier. This time frame overlaps with an estimated domestication age of 4,800 BP achieved by DNA studies. Nevertheless, the association of the earliest domesticated finds with pastoralism is clear, and this suggests that pearl millet domestication may have taken place amongst forager pastoralists. Of potential importance is that pearl millet constitutes a poor forage crop as growing grass, the grains being protected from grazers by a bristly inflorescence. This represents a contrast to sorghum, which makes an excellent and preferred source of fodder. Thus, livestock would have been detrimental to early sorghum cultivation in contrast to pearl millet cultivation that had inherent protection from overgrazing. Along similar lines, Mercury and colleagues have suggested that early cultivated millets of the central Sahara in Libya, such as Echinochlua colonna and Panicum laetum, lend themselves to domestication because of weedy traits such as resilience to grazing. Thus, sorghum and pearl millet, despite similar chronological and climatic circumstances of domestication, appear to have been influenced by differencing socio-economic ecologies, especially in terms of the compatibility with pastoralism. Once domesticated, both sorghum and pearl millet were integrated with pastoralism, providing a potent economic package for dispersal. Pearl millet's earliest and most rapid spread was perhaps eastwards across the Sahara, reaching eastern Sudan by probably 1850 BC and India via maritime links by about 1700 BC. The archaeological record is more robust when it comes to spread within West and Central Africa, where pearl millet made steady progress southwards into ever wetter and more tropical ecologies, eventually extending into openings within the rainforest zones by about 400 BC. Estimates of the land area covered by the dispersal of sorghum and pearl millet farming over time is remarkably similar, suggesting that similar processes of dispersal, that is, serialization, took place by growing agropastoral populations. In addition to early Sahelian dispersal and long-distance translocation to India, both crops spread southwards in Africa, cutting across different climatic and vegetation belts at gradually increasing rates.